Nearly 2 centuries, doctors have used anesthesia to safely render patients unconscious during surgery. Yet the exact molecular mechanism behind that profound effect remained elusive. Why do these drugs work so reliably, and what happens in the brain when consciousness fades away? Today we explore a breakthrough discovery that finally sheds light on the molecular secrets of anesthesia-induced unconsciousness, unraveling a mystery that has puzzled scientists and clinicians for generations. The heart of this discovery are sodium-ion channels, tiny protein structures embedded in cell membranes. These channels regulate the flow of charged particles, enabling neurons to generate and transmit electrical signals. Without these signals, communication between brain cells grinds to a halt. Researchers have long suspected that anesthetics target these channels, but the details of this interaction have been difficult to capture, especially in complex mammalian brain. Hailed anesthetics like Sevoflurane are widely used in operating rooms around the world. These drugs suppress brain activity, producing unconsciousness and immobility. But how do they achieve this at the molecular level? The answer, as revealed by recent research, lies in their subtle yet powerful interaction with sodium channels, effectively dampening the electrical chatter that keeps us awake and aware. A team of researchers from Weill, Cornell Medicine and Birkbeck, University of London, joined forces to tackle this mystery. Their work, published in Nature Communications, provides the first atomic-level view of how sevoflurane interacts with sodium channels. This collaboration combined expertise in anesthesiology and structural biology, paving the way for a deeper understanding of how anesthesia works. Visualize the anesthetics effect. Scientists turn to a marine bacterium, Magnetococcus marinus. Its sodium channels are structurally simpler than those in humans but function in a remarkably similar way. This simplicity allowed researchers to capture detailed structural snapshots, offering a unique window into the molecular dance between anesthetics and ion channels. Using high-resolution X-ray crystallography, the team captured images of sevoflurane bound to the sodium channel. They discovered that the anesthetic fits into a small pocket at the edge of the channel's pore-forming region. This binding site is away from the main pathway for sodium ions, but it plays a crucial role in stabilizing the channel in an inactive state. When sevoflurane binds to this pocket, it makes the sodium channel less likely to open. As a result, sodium ions can't flow as easily, and neurons struggle to transmit electrical signals. This silencing of neural communication is what leads to the loss of consciousness and immobility during anesthesia, a direct molecular explanation for a phenomenon observed for generations. To test their findings, researchers altered just one amino acid in the binding pocket. Remarkably, this tiny change prevented sevoflurane from binding effectively, and the channel no longer stayed in its inactive state. This experiment confirmed the critical importance of the binding site and highlighted how even small molecular changes can have dramatic effects on anesthesia. While the study used bacterial channels as a model, the ultimate goal is to understand how these findings translate to human biology. The researchers are now investigating whether similar binding sites exist in mammalian sodium channels. If so, this could explain why some people respond differently to anesthesia and open new doors to personalized medicine. Understanding the precise molecular mechanism of anesthesia could revolutionize drug design. With this knowledge, scientists may be able to develop new anesthetics that are safer, more selective, and have fewer side effects. This could make surgery even safer for millions of patients worldwide and reduce the risk of complications. Beyond anesthesia, these findings offer a rare glimpse into the biology of consciousness itself. By studying how certain drugs can switch off awareness, scientists hope to better understand what it means to be conscious and how the brain generates our sense of self. This research could have profound implications for neuroscience and philosophy alike. For anesthesiologists, understanding how these drugs work is more than academic. It's essential for patient safety. When patients react unexpectedly to anesthesia, doctors need to know why. This research empowers clinicians with deeper knowledge, helping them troubleshoot problems and provide the best possible care during surgery. The journey doesn't end here. Scientists are now searching for naturally occurring mutations in human sodium channels that might affect anesthetic binding. Such discoveries could explain individual differences in anesthesia response and lead to new insights into the genetic basis of consciousness and drug sensitivity. This breakthrough marks the beginning of a new era in anesthesia research. With atomic-level detail, scientists can now design better drugs, predict patient responses, and perhaps even unlock deeper mysteries of the mind. The future of anesthesia is brighter and safer than ever before. Summary: the discovery of how anesthetics like sevoflurane bind to sodium channels and induce unconsciousness is a major leap forward in medical science. This knowledge not only solves a long-standing mystery, but also promises to improve patient care and inspire further research into the workings of the human brain. As we continue to explore the molecular foundations of consciousness, the possibilities for medicine and science are truly exciting.